Our next speaker, our final speaker, is Dr. Michael Skinner, a PhD, who is a biologist specializing in epigenetics. He's a professor in the School of Biological Sciences at Washington State University and the author of a landmark study, Ancestral Dichlorodiphenyl Trichloroethane, that is DDT exposure, promotes epigenetic transgenerational inheritance of obesity, which determined that exposure to the insecticide DDT impacts multiple generations, uniquely contributing to obesity 3 generations down the line. He did his bachelor's in chemistry at Reed College right here in Portland. His PhD is in biochemistry at Washington State University, and his postdoctoral fellowship was at C.H. Best Institute at the University of Toronto. Dr. Skinner's research is focused on the investigation of gonadal growth and differentiation, with emphasis in the area of reproductive biology. His current research has demonstrated the ability of environmental toxicants to promote the epigenetic transgenerational inheritance of disease phenotypes due to abnormal germline epigenetic programming in gonadal development. Dr. Skinner established and was the director of the Washington State University and University of Idaho Center for Reproductive Biology since its inception in 1996. Dr. Skinner has received all kinds of awards and recognitions. He is internationally known and very recently has come to be in a very unique international group which is undergoing the process that I refer to as silencing of the lambs, which includes Tyrone Hayes and Judy Carman and other people, where there's an increased effort to silence the people who provide information that do indicate that our health and our future health over multiple generations is likely in jeopardy. Dr. Skinner.
Well, thanks very much. I want to thank Jay and Warren and the organizers for the opportunity to be here. So what I'm going to talk to you about is how is it that the exposures we've been talking about actually cause disease? So in other words, the mechanism. So when you get the exposure, what happens to then later in life, how are you going to get the disease? And then I'll talk about how it's not going to only affect you, but what your great-grandmother was exposed to may cause you to get a disease even though you've never seen the exposure, and you're going to pass it on to your grandkids. And so this actually will be a little bit of scary stuff, but believe me, in the end, I'll actually turn it around and show how we can use this information to significantly impact our health in the future. So what I'm going to talk about is the concept also, not only this transgenerational, that how you pass it to the next generation and how that's influenced by these environmental chemicals, but this concept called epigenetics. And so I'll actually describe that during the talk, so when you finish, you should have a pretty good understanding of that. And what these environmental or ancestral ghosts in your genome are, pretty much you'll understand that. So I'm going to do something different than maybe your normal talk. I'm actually going to show you some data, because I don't think it's appropriate for me just to make a comment without having the data there. Now, you may not be able to interpret the figure, and I will explain it, so don't worry about it. But I really think it's important to understand that there's data to back up everything I'm going to talk about. Okay? Because this is sort of at the forefront of an area that we don't know a lot about. Okay? So, as a preamble, in science, there's a concept called, and the most predominant concept today, is called genetic determinism. The idea is that the genetic sequence, the sequence of the DNA, is the building block for all of biology, which it is. And that basically it determines everything that's going to happen. So if I pull you off the street and say, where do you get your disease? more than likely you will tell me it's some genetic mutation that you inherited or obtained that's going to cause that disease. If I ask you, how does evolutionary biology work? In other words, the evolution of a species. More than likely you will tell me it's random DNA sequence mutations that cause a change in the species. This genetic determinism is basically so infiltrated in terms of science thought that we don't think anything outside it. That's the only thing we think about. So what I'm going to actually tell you is genetic heresy. Essentially, what I'm going to show you is it's not this flat concept of genetics, but it's this round concept of a combination of epigenetics and genetics. This is probably, it's that degree, whether the earth is flat or round. That's what we're actually talking about in terms of a scientific concept. Because this is, for 100 years, been ingrained in our society so much. The key thing is, this mechanism I'll discuss gives us whole insights in terms of how things like environmental exposures can actually cause diseases to happen. All right? Now, some of the things that sort of led us to this is not just out of vacuum. If you go, and here's just a number of different examples of environmental impacts on biology. Every region of the world has unique disease frequencies. So in Japan, they have very high rates of stomach disease, extremely low rates of prostate disease. North America, we have very high rates of prostate disease, really high rates of prostate disease, and very low rates of stomach disease. If you take someone from Japan early in life, before the age of 5, and move them to North America, guess what? They developed a North American phenotype. I'm sorry, this can't be genetics. It has to be an environmental influence on the disease. If you take pretty much any disease that's been looked at, prostate cancer, mammary cancer, breast cancer, Alzheimer's, all sorts of different diseases they've looked at. There's been a program called Genome-Wide Association Studies that the NIH has supported, where they actually take 1,000 patients with a given disease, and they'll look for genetic mutations in that population to give them insights on what's causing that disease. And for many diseases, they've found correlations with these genetic mutations, these SNPs we just heard about. But the problem is, it's generally in less than 1%, 1 or 2% of the disease population. Thus, for breast cancer, for example, it's less than 2% that actually have a genetic mutation in the whole population of breast cancer patients that you can put your finger on. Now, to a degree, they say, well, we just haven't looked hard enough. We need to look harder in terms of what's going on. What I would sort of say is, maybe it's something else besides just genetic mutations. And it's not that the genetic mutations aren't really critical. It's just there's other things going on. If you look at most any disease, just pick one, Alzheimer's, or better, let's say, autism. Over the past 25 years, many of the diseases have been increased over 10-fold. I'm sorry, genetics can't explain that. There's no genetic mutation that can explain that rapid of increase. That has to be an environmentally induced phenomenon. If you take identical twins that have almost the same genetics, when you have them grow up, guess what? They develop different diseases. If it was all based on genetics and only the DNA sequence, they should get the same diseases. Again, the environment is influencing those diseases become. There's hundreds, maybe thousands of environmental exposures and compounds that are directly linked to disease. Believe it or not, the vast majority of those cannot change the DNA sequence. Most of the compounds we've discussed in the last day and a half cannot mutate the DNA. Mutagens are very, very rare, although there's some. The vast majority of things can't change the DNA. The DNA sequence is extremely stable. It's very difficult to actually cause that mutation. So if that's the case, why is it they're actually promoting things if it's just genetics? Okay. And I won't talk about this in detail, but we have the data around evolution as well, rapid evolutionary events, that also fit very well in what we're going to talk about. So what it looks like is there's something, it's not the DNA sequence, and genetics isn't really critical. It is a hallmark of science. The problem is, more than likely there's something else that's regulating how that DNA functions that's independent of the DNA sequence. And so what I'll discuss is this concept called epigenetics. And epigenetics actually provides solutions for a number of those failures of genetic determinism. And again, I want to stress, it's not the genetics and DNA sequence isn't really critical. It's just a piece of a much bigger story. And for geneticists, for the past 100 years, we've all been taught this. It's a difficult thing to think about something beyond just the genetics. Okay? So I'll stop preaching now and actually move on to the talk. So what I'm going to talk about is epigenetics and disease phenotypes. What I'll mention is how environment can actually influence the epigenetics to then later in life promote a disease. There's a direct link, and I'll discuss that. There's a concept that's developed over the past 10 years in medicine called the developmental basis of adult onset disease. Believe it or not, what's happening when you're 60 and you get breast cancer, it's not what's going on right then when you're 60. It was what happened back when you were in puberty. When your mammary gland was first starting to develop, the exposures at that time more than likely influenced your ability to get the cancer later in life. This has been shown with a whole variety of different diseases and so forth. So this developmental basis of disease, the problem has been, how does that work? If something going on here that's affecting the genetics is just a transient thing that then goes away, why does it affect something 30 years later? Well, epigenetics provides that mechanism. And then I'll talk about this very heretical sort of thing called non-genetic inheritance. Right now, if you think about what have you been taught in terms of what you're inherited, it's all genetics. It's all your DNA sequence. What I'll talk about is a concept that you can actually inherit things through epigenetic mechanisms that have nothing to do with the sequence. So it's a form of non-genetic inheritance. Okay? And it explains a lot of the things we've seen in inheritance, why a disease is not following normal Mendelian genetics, why it actually is familial in the family, but doesn't follow the actual genetics. Okay? We can explain why that happens. So we'll talk about this concept of epigenetics, and I'll describe it during the talk. So around 10, 15 years ago, we were doing a study just looking at the effects of environmental compounds, and I'll discuss a number of them in a minute. And we use we use rats as a model, because you can't really do this effectively on a human where you have an experimental model. So we exposed a gestating female rat at a critical time of development called, it's when your gonad is turning into an ovary or a testis. It's called sex determination. When the sex is initially being established, it's in the first trimester of fetal development, okay? So we exposed that then we got an offspring. We got that offspring and we bred it for 4 generations. The only exposure was the gestating female originally. So this is called F0. First, the fetus that was actually exposed is called F1. Then you have a grand offspring, F2, great grand offspring, F3, and so forth. So I got out 4 generations. The first compound that we actually studied extensively, which we've now studied a series that I'll talk about, is called vinclozolin. Vinclozolin is an agriculturally used fungicide. Particularly, it's probably one of the biggest ones used in the fruit and vegetable industry. It's a very effective fungicide. And this is basically shown in the wine industry. Don't panic. It turns out its half-life in water is actually fairly short, so it doesn't ever end up in the wine that you drink, okay? Okay. But during the occupational exposure and so forth, it's actually an issue. So it turns out that this fungicide is an anti-androgenic substance, meaning that it binds to the androgen receptor and it blocks the actions of androgen. So it's an anti-androgenic. So we chose it for that purpose, actually. So what we found is if we expose this gestating female, the individuals born, the F1 generation, exposed as a fetus, as they grew up, we had control animals that were injected with a vehicle, and then vinclozolin was the treated animals. What we found is that vinclozolin was present. We have high rates of cell death in the testis. The cells that are going to turn into a sperm started to die at high levels. Okay? And that was in the F1 generation. And so that's effectively the first phenotype that we found. And then what we found is, when we bred the animals, 90% of the males, as they became adults, had this abnormal germ cell death. For first generation, the second generation, the third generation, fourth generation, constant, okay? And then if we outbred the males, we maintained the phenotype. If we outbred the females, we lost it. So therefore, it's being passed through the sperm, not the egg, okay? So it's going through the male line. This particular one is. And here's the controls that didn't have this. Now, normally what you would see in genetics is every time you brought in new genetics in terms of breeding, you would see a decline in the frequency. So after 4 generations or 5 generations, you'd actually see a decline to where you had very little frequency. So this is following at what you would call a non-Mendelian genetic phenomena, which has been shown, hundreds of cases of this have been shown in a number of different systems. Now, as we allowed the animals to grow up to be a 1 year of age, which is about a middle-aged sort of rat, 17% of the males develop mammary tumors. Normally, they don't at all. This also occurred in the females as well. Over 50% of the animals develop prostate disease. If you don't know, in the human population, 50% of the human population over the age of 50, males, have prostate disease. It could be just benign, but it's basically they have prostate disease. Almost 100% over the age of 70, so this is really a most prominent disease. We had about 40% that had kidney disease. You could see cysts developing in the kidney, and they're actually blood markers we use to actually look at this as well. The testis had 30 degrees, so as you became an adult, younger you had fertility, but as you became an older adult, you lost your fertility, okay, about 30%. Immune abnormalities in about 30%. So this, what this showed us is about 90% of the animals had at least 2 or more diseases. And so this was a very, very strong disease phenotype being passed for 4 generations in a non-Mendelian sort of manner. So then, was this something that was just unique to vinclozolin? So we actually took and looked at the compound specificity. Now, if you don't know, one of these experiments takes about 3.5 years. Okay, so this is not a quick thing, and it's extremely expensive. So we actually looked at these, 8 different compounds here. Vinclozolin we repeated. Flutamide, which is a pharmaceutical used as an anti-androgenic substance to treat prostate disease, was used. We get this repeated. We have the F3 generation effect and a F1. Interestingly, the flutamide did not go transgenerational. It just affected the first generation, then it stopped. So it's not just the anti-androgenic part of this. We use dioxin. Dioxin is an industrial contaminant, and this basically caused an F3 generation effect. Plastic, the bisphenol A and the phthalates, the things in the plastics that we drink out of all the time, caused an F3 generation effect. Jet fuel, hydrocarbon, this is just essentially like diesel, caused an F3 generation effect. The pesticides most commonly used in humans are permethin and DEET. Okay? Interestingly, they did not cause an F1 generation effect, but did cause an F3 generation effect. Okay? DDT, okay, which is I can discuss later. This is not, even though it's banned, about 5 years ago, the Gates Foundation pushed its use in Africa and for an anti-malarial sort of thing, and now it's heavily used in India, Africa, several countries in South America, and France. So this is now gaining use again. And this also caused a transgenerational effect. And the methoxychlor, which was the replacement for DDT, also does as well. Okay? So turns out that a number of these compounds, the dioxin, plastics, jet fuel, and pesticides, were actually supported by the Department of Defense because they were interested in military exposures, particularly in the Gulf Wars. Highest one being hydrocarbons because they spray the roads with jet fuel to prevent dust. Surprisingly, from this observation, they're now removing that. They're not spraying so much jet fuel in the compounds and so forth. So they actually supported financially this. So I'm just going to show you 2 examples of the disease before we move on to the epigenetics. One of the most common reproductive problems in females is polycystic ovarian disease. In certain populations, it's 18% of the females. It's definitely over 10% in most of the populations in developed countries. So this is where the ovary develops these really large cysts. It influences their reproductive problems, causes pain, and influences their fertility as well. So this is the #1 reproductive issue in women today in terms of reproduction. If you looked at the F1 generation, only plastics promoted polycystic ovarian disease in the F1 generation. Okay? But if you look at the F3 generation, the great grand offspring, 3 generations removed from the exposure, essentially all of the compounds we tested promoted polycystic ovarian disease. Okay? This also had endocrine abnormalities that were correlated with it and so forth. We've seen an increase in polycystic ovarian disease over the past 20 years from a few percent to over 10%. Okay? So what if these ancestral exposures might be a big component in why we're seeing this increase today? Now, this is the one that Warren mentioned. It's in the DDT. And so we did a DDT exposure. In the F1 generation effects of both males and females, there was no obesity. There was no effect. They did have a few disease phenotypes, but no obesity. In the F3 generation, for both males and females, over 50% of the females and 50% of the males in the population developed obesity. And interestingly, it's really a susceptibility for obesity because two animals, one developing obesity and one's not. So this one's susceptible to it and this one's not on exactly the same diet, exactly the same exercise and so forth. This one will develop obesity and this one won't. So we've induced the susceptibility under a certain diet regime and so forth for you to get obesity. It's not that we promoted the disease, it's susceptibility. And here this shows in the abdominal area how the fat deposition in the abdomen is essentially on every single organ in the obese animal, but it's relatively normal for the non-obese. So what if, in the 1950s, when we essentially for 10 years had exposure to DDT, and essentially in the North America, every single person got exposed to DDT in the 1950s. It was so heavily used. We are now 3 generations from that population. In the 1950s, the obesity rate was less than 3%. Today, it's 35% in the United States, and it's on a rapid increase. So could these ancestral exposures actually be influencing these generational effects on disease phenotypes we've seen? Which is something we need to think about. Not that disease, or not that diet, exercise, and all those sort of things are really critical for the onset of the obesity and metabolic syndrome, in terms of whether you're going to get it or not. But the susceptibility could be there from something like this. Okay? So, and if we look at disease, we have reproductive defects in the female and the male, kidney disease, prostate disease, a whole series of brain defects I didn't have time to go into in terms of behavior, anxiety behavior, mate preference, preeclampsia, which is a major problem with females, there's polycystic ovarian disease and obesity, and the percentages are actually fairly high for many of these diseases. This is not a trivial disease induction. And this is actually what's going on 3 generations away from the exposure. Okay? So if you expose a gestating female, the F0 generation mother is being exposed. The fetus that's going to turn into the F1 generation is being directly exposed. And the germline that's inside that fetus that's going to determine the grandchild is also directly exposed. So if you have to go out to the F3 generation to have a situation where there's no potential chance of exposure. So that's why we always deal with the F3, the great-grandchildren, actually to get away from any potential chance of direct exposure, because we can't rule out these sort of things. And so that's the case with any species. All right. So now I want to talk about the mechanism. How does this work? Okay, and as I mentioned early on, this wasn't following sort of normal genetic phenomena. If this was a DNA sequence mutation issue in genetics, the frequency of induction of a DNA sequence mutation with things that can do it are actually 0.001% of a population. It's extremely low. Even in a hotspot, it's only 1 or 2%. Okay? The problem with this is it's extremely random. You can't promote the same mutation in the DNA at the same spot every time. In fact, it's very random throughout the genome every time it happens. So therefore, you would never get the same phenotype. You'd have these random phenotypes all over the place because of the random nature of DNA sequence mutations. This would follow normal Mendelian, so we should have seen a decline in frequency at each generation. So this phenomenon that we were looking at really didn't fit this concept of genetic. But we looked initially because that was what we were taught to look at that. So we looked very carefully. We did some experiments, and we found in the first generation, we found no evidence of any genetic mutations looking for either point mutations or copy number variations or other types of mutations. Okay? Another mechanism is called an epigenetic mutation. Okay? Now I'll describe that in just a second. Epigenetics can occur in exceedingly high percentages. 30 to 100% is not uncommon for an epigenetic phenomena. These are highly reproducible. The same site gets affected every time. So you get the same phenotype. This does not follow normal Mendelian genetics. And so it looked like what we were looking at was actually epigenetics. Okay? Now, So epigenetics is defined or described as molecular factors or processes. These are molecular sort of components around the DNA. So they physically are around the DNA. Okay, so they exist associated with DNA. And they can regulate genome activity. And that means what genes are turned on or off. If the epigenetics didn't actually regulate what genes are turned on and off, it would be irrelevant. It has to be able to regulate the genes going on and off, otherwise it has no effect. Okay? So that's part of it, is it needs to regulate what's called genome activity, what genes get expressed, or what genes are turned on and off. This has to be completely independent of DNA sequence. It doesn't require any specific DNA sequence to have it function. If it did, it would be genetics. Okay? So this is completely independent. It doesn't matter what the sequence is. It's a much more of a sort of a different sort of a mechanism. Now, and probably most importantly, this is what's called mitotically stable. So when a cell divides from a parent cell into two daughter cells, and it grows, it divides into two cells, that's called mitosis, or cell growth. So when the cell actually grows into a daughter cell, two daughter cells, the cell has to completely replicate the epigenome that was in the parent cell. So all of the things that are going on in the parent cell have to be replicated and appear in the daughter cell. If it didn't do that, it was only just in the parent cell and then disappeared, again, it would be irrelevant. It has to be what's called mitotically stable. Just like the DNA is replicated, the epigenome is replicated too. Okay? So this is the definition. So it's those molecular factors around DNA, chemical modifications and so forth, and I'll describe in a second, in terms of how regulating how that DNA functions. Okay? It's independent of DNA sequence, and it's stable. So therefore, let's say you were in puberty, and a female, and you were exposed to an environmental chemical. The chemical actually went in and changed your epigenome in your early developing mammary epithelial cell, and shifted that epigenome mark, or a series of marks. As that cell divides through the next 30 to 40 years, it's going to maintain those epigenetic marks. Thus, when you're 30 or 40 or 50, you have an increased susceptibility to develop the breast cancer. So this is how an early event in life can be maintained for decades and cause a disease later on. Okay? So there's several different kinds of epigenetics. It's not critical to know them all, but I just wanted to give you the concept that there's a thing called DNA methylation, which I'll discuss in a minute, where there's a chemical modification of the DNA. There's a histone. The DNA actually has a bead on a string sort of thing, where there's proteins that form a spherical sort of structure, and the DNA gets wrapped around it. And then there's another one, and then there's another one. So you have this beads on a string type thing. Okay? Those proteins can be chemically modified as well, and that can change how the genes are expressed up and down, independent of the DNA sequence. So that's epigenetics. There's another way in terms of the structure of the DNA, whether it's coiled, looped, stuck to the matrix. There's all sorts of structural sort of things. Those structural things are also epigenetics, and they can change what genes go on and off independent of sequence. And then there's a series of what's called non-coding RNAs. These are products of the DNA, this RNA that's product expressed, that doesn't make into protein, but actually has other functions that's independent of DNA sequence. So all of these are very critical epigenetic components. Turns out the phenomena we were studying was going through the sperm, and so this is called spermatogenesis. All of these are irrelevant except for DNA methylation. So therefore, we concentrated on DNA methylation. Okay? So here's your genetic sequence, your nucleotides that are there. There's an enzyme that actually puts this methyl group on what's called a cytosine. So that small methyl group is an epigenetic mark. Okay? And that epigenetic mark can actually change how the DNA functions. So this C sits next to a CPG, and then as the cell divides, undergoes that mitosis and grows, it's going to replicate this DNA sequence. Well, it turns out there's an enzyme sitting there called methyltransferase. If it sees the methyl group on the parent strand of DNA, it'll methylate the new strand of DNA. So thus you replicate your epigenetic mark as the cells grow. Okay? So this is DNA methylation. So we should have actually known this before, But it turns out during development, there's a couple of times, believe it or not, when the DNA methylation is completely erased, or nearly completely erased, and then returns back to normal. So the first time this happens is during fertilization. When an egg and a sperm come together, the DNA immediately gets demethylated. And this is what creates what hopefully everybody's heard about, called an embryonic stem cell. The reason the embryonic stem cell is totipotent, it can turn into different things, it doesn't have any DNA methylation. So there's no driver in terms of its differentiation. So it's completely totipotent. Then as the embryo starts to develop, it's remethylated in a cell-specific manner. So this has been known. Except for a small set of genes called imprinted genes that maintain their methylation. It's not erased, and so therefore it gets passed from generation to generation. And they're very important genes. The other time is during that time when we did the exposure at sex determination of the fetus in the early stage of fetal development. There's this demethylation of that germ cell that's going to turn into a sperm or an egg, then a remethylation at sex termination in a female or male specific manner to give you a sperm or an egg. So we did this exposure here. We essentially remodeled the epigenetic programming of the germline. It became permanently programmed, imprinted-like. It gets passed from generation to generation. Thus, a non-genetic form of inheritance. Okay? All right. So the next question was to sort of actually experimentally show that this was going on. So this is just a method that we use. What we do is we fragment the DNA, we pull down the methylated DNA, and then we put it on what's called a high-throughput sequencer, sort of a high-tech procedure to actually map where the epigenetic sites are. And when we do that, here's just all your chromosomes from chromosome 1 to 20. Here's the size of the chromosome here. And so this is actually you're looking at the whole genome. And wherever there's a red arrow is where there's an epigenetic site we identified in the F3 generation sperm for this exposure. If the arrow is going down, it's a decrease, and if the arrow is going up, it's an increase. Okay? So we actually map the epigenome of the sperm, showing that it was permanently affected for generations to come. This is F3 generation sperm. Okay? We've done this now with everything else we've looked at, all those other compounds. And when we look at all those other compounds, pesticides, hydrocarbons, dioxin, plastics, here's vinclozolin, each one of the epigenetic marks in those compounds, if you look in the middle, there is no overlap. So it turns out we have exposure-specific epigenetic marks for each exposure. So think about that. Would there be a way to actually diagnose what you were exposed to early in life, or what your ancestors were exposed to, if there's an exposure-specific mark for the specific compounds? So this could really improve our ability to do toxicology as well as sort of diagnose. And so that's something we're excited about, plus something we did not expect at all. So we have these epigenetic biomarkers for ancestral exposure. That most of them are not in promoters, they're away from promoters. And the next question was, how do these marks affect what genes are turned on and off, the gene expression? So what we did is took 11 different tissues from males and females, took those tissues out and looked at what genes were being expressed, which genes were on and which genes were off. It turns out generally each tissue has a tissue-specific expression profile, certain genes that are present or absent. When we did this in terms of the transcriptomes of the F3 generation from, let's say, the female, the heart, uterus, ovary, liver, kidney, we found, let's say in the heart, there were 405 genes that had an altered expression because of this F3 generation vinclozolin exposure. Okay? And the uterus had nearly 200, and the liver had 99. So then when you looked at all of those, again, when you looked at those expression profiles, there was none that were overlapping. Same thing was going on in the male. Okay? So what we had was over 5,000 genes that were affected, but we knew there was probably only a couple hundred epigenetic changes. So how does a couple hundred epigenetic changes regulate 5,000 genes in a tissue-specific manner throughout the body? Okay? So it turns out if you took those genes and you clustered them on this genome, remember that here's your chromosomes, here's the size. For a male or a female, the females are green and the males are red. Many of them clustered together, but there were clusters of genes. They were all starting to cluster together. When we look farther, it turns out there's an epigenetic site, one of these what we call epimutations, a differential methylation site, that sits in a region that gives you, let's say, for example, this is like 2 to 5 megabases. The way to think about that, a normal gene is only a few 100,000 bases. So in this region, we could have 50 genes in one region with one epimutation, and this now, through this epigenetic mechanism, can regulate all 50 of them. So what I'm saying is subtle shifts in the epigenome have an ability to dramatically influence genome activity, or what genes are turned on and off, in a cell-specific manner. So this is truly a sort of a non-genetic approach, or a view of how the genome's actually regulated. Okay? All right, so we had these, what we call epigenetic control regions that we identified. All tissues and cell types we've looked at actually have a difference. So in other words, if the embryonic stem cell, or the sperm or the egg, is carrying this with it, an embryonic stem cell now has a shift in its epigenome, every cell that comes from that embryonic stem cell now has a shift in their epigenome. Thus, when it becomes an adult cell, every single cell type has a shift in what genes are on and off. So this dramatically influences the whole organism. Okay? So, now I want to just give you one example of a disease that's developing. So here's an ovarian disease, that polycystic ovarian disease. So here's a cross-section of an ovary where you have small follicles developing into large follicles that eventually ovulate. And there's a cell inside this developing follicle. Here's the oocyte shown here, the egg. There's the cells around that called the granulosa cell. Granulosa cells are really critical to support and provide nutrients and signaling for that developing egg. So we purified or pulled out the granulosa cells from these animals, and the control and the enclosed one. Then we looked at the genes. We found there were 500 genes that had an altered expression, being on or off, in the granulosa cell compared to the control, in the F3 generation granulosa cell. And when we looked, a large portion of those genes had been previously shown to be linked to ovarian disease, specifically polycystic ovarian disease. So what we did was this generational effect through the epigenetics altered this permanently, so that the granulosa cells now have this disease susceptibility built into them. So that's how the germline transfers this to a disease in an adult animal. Okay? Okay. So the mechanism for this environmental epigenetics is that the environmental factor affects this gestating female, where there's the developing embryo and fetus. At this time of sex determination, when there's a reprogramming of that DNA methylation, it gets altered and permanently programmed, so that when that animal becomes an adult, the F1, the sperm or the egg, if it's a female, passes this germline differential methylation site to the next generation. All of the tissues in that animal will be altered in terms of what genes are on and off. Tissues that are sensitive to that shift have a susceptibility to develop disease. When this animal reproduces, the same program is in the germline, it gets transferred to the next generation, and so forth. So this is a form of non-genetic inheritance, which in contrast to genetics is very sensitive to environmental insults. Okay? And so this is this epigenetic transgenerational inheritance and this form of non-genetic inheritance. So this is a transient embryonic exposure at the time of sex determination. This provides this fetal basis of adult onset disease we talked about earlier. A whole series of diseases affected in the brain and so forth. Now, there's a concept called germline, that's the sperm or the egg, transmission. That's called a transgenerational phenomenon. But the majority of cells affected from an exposure are called somatic cells. Every cell in the body is a somatic cell except the sperm or the egg. Okay? So the majority of exposures are on your somatic cells. So when you're in puberty and your mammary gland gets exposed to an insult, the somatic cells of the mammary gland are going to get affected. So when you're older, you have a susceptibility to develop breast cancer because of that. That's a somatic cell effect. You can't transfer that to the next generation. Okay? That's just a somatic cell direct exposure. However, the mechanism for those direct exposures is exactly the same. In an early stage of development, you have this cascade of genetic events from that early stage of puberty for the mammary epithelial cell is then going to go at this cascade of genetic events where the gene expression slowly goes through a cascade of events to reach a normal differentiated or developed state. The epigenetics is doing the same thing. You have this combination of epigenetics and genetics working together to give you this normal methylation state up here that gives you the normal gene expression. Now, if you have an environmental insult over here early in development, like during puberty, you shift that epigenome, all of a sudden the whole thing gets shifted so that you end up with an environmentally modified epigenome and gene expression profile of what genes are on and off. This modified gene expression profile increases your susceptibility to get disease. You're not going to pass that on, but that somatic cell effect is through the same type of mechanism that we talked about for the generational thing. Okay? All right. So this phenomena is called epigenetic transgenerational inheritance. All right? It's a non-genetic form of inheritance. It's involved this epigenetics. For toxicology, all of the compounds that we used were used at higher levels than what you'd expect in the environment generally. Some of them were environmentally relevant, but most of them were a little higher because we were studying the phenomena. We weren't doing risk assessment, toxicology, okay? So I don't want to say anything about the risks of these things, but all of them had the capacity to promote this phenomena. So in toxicology, if it's not just the individual exposed that's a problem, but the generations to come, that sort of increases the biohazards of these exposures. And so we need to think about that in terms of toxicology, and that's something that's not done effectively today. Okay? We permanently changed the germline of the epigenome. So in other words, what this did is it affected the progeny for generations to come, and it can't really get fixed. It's basically permanently programmed. So it's almost like an evolutionary event. We shifted this sort of expression profile and phenotypes permanently. Okay? So that's something we need to think about, plus it tells us a lot about how biology works. We think that this epigenetics will be a very critical component of where disease comes from, what's called disease etiology, which we simply have not paid attention to because we've been focusing primarily on genetics. It's not that the genetics won't be a really important component, but the two of them sort of are integrated, and we need to look at both. For evolutionary biology, this is also fairly critical. If you have a population that's exposed to a certain exposure, now it turns out it's not just chemicals. Nutrition will do the same thing. Whether there's, in a human population for the Dutch famine and a Swedish famine, caloric restriction, they've found generational effects because of that, the caloric restriction, okay? And also stress, high stress, particularly during pregnancy, affect the fetus such that there's brain programming and so forth, and in the germline going forward as well. So nutrition and stress can do this as well, not just environmental chemicals. So if you had a population for evolution that was exposed versus not, and they stayed separated and had a continued sort of different environmental exposures, essentially what you would do through this type of mechanism is drive a potential speciation evolutionary event. Okay? So in terms of the epigenetics, you affect this genome, you affect the expression, you express the physiology within the individual. That then can promote an abnormal biology or a disease onset. If it's a population-wide thing, and now you have a phenotypic shift, a trait shift in the population, such that natural selection can act on that to select that trait, essentially you're going down an evolutionary pathway. So this is important not only for sort of normal disease physiology, but also evolution as well. Pretty much every area of biology. So in the last slide, the environment has the ability to influence this epigenetics in contrast to genetics. It can't really change the DNA sequence, but it can change these epigenetic marks that we talked about. This has dramatic effects in how the genome functions. So if these marks or epimutations get put in there, this is your ancestral epigenetic ghost that's in your genome. Okay? This then affects what genes get turned on and off through this transcriptome, these genes that are on and off at the time. That can give you a normal or abnormal physiology, thus promoting disease. And because it's in the germline, it essentially gets passed from generation to generation. Now, what I talked about initially was through the sperm, But it turns out recently, let's say we did methoxychlor, it's all going through the female line. DDT goes about 50-50 through the male or female. So it's not just through the sperm, you can also have an effect on the egg as well. Okay? Now what I will finish with is, this occurs in insects, like basically flies, worms, plants, mammals, a number of different kinds of mammals, fish. This is across the board, all species that have been looked at. So what's going on in your honeybee, if you don't know this, the reason a queen develops versus a worker bee is the queen was exposed to a thing called royal jelly early in development. It promoted that queen to develop. If the workers didn't get exposed to it, they develop as workers. The basis for that, epigenetic regulation of the genome. So therefore, the same thing's going on in an insect that's going on in a human. Okay, so this is not just you need to think about this globally, not just for our particular health. All right? Okay, so with that, I just want, and then here is the positive sort of thing. Yeah. So it is pretty negative to think about these generational effects and how this is working. But the flip side is, just by knowing this exists, now we have a whole series of things we can do which we couldn't do before. We never knew how these things developed and knew why disease developed and so forth, okay? So this is what's potentially possible. When you're 20, you go in and get your epigenome mapped. Because of those signatures of the exposures, we can determine whether you or your ancestors may have been exposed to something, specific chemical, that then is directly linked to a disease that's developing later on, okay? So by simply knowing that's the case, then in your 30s or 40s, if let's say you have a susceptibility for breast cancer or prostate cancer, we do a therapeutic treatment, which there's actually a number of them known today, that could prevent the disease from developing. Not treating it after it developed, but prevent it from developing it in the first place. This is called preventative medicine. And the reason we haven't been able to do it is we didn't have those early stage diagnostics. Genetics was thought in 2000, when we actually sequenced the genome, it was thought that genetics might tell us this. But unfortunately, looking very hard, genetics just didn't have that flexibility to give us those types of marks, only in small percentages of the population. Epigenetics may be much more efficient at doing that. So by simply knowing, and these are the things that now newer people following up our stuff is starting to look for. Can we actually do these early stage diagnostics for exposure and disease? So the flip side is, I think this will significantly enhance healthcare in the future. Not to say we shouldn't try to avoid as many exposures as we can. So with that, I'll finish, and happy to answer any questions later on.